Hi all, thank you for joining my poster session. I am Melike Karakaya from Turkey, affiliated to Biruni University and Bezmi Vakıf University. I will talk about a small preliminary study on delineating highly conserved HLA supertype-restricted epitopes in the SARS-CoV-2 structural proteins. The current ongoing COVID-19 pandemic demands a better understanding of the host-pathogen interaction. Though there are already vaccines against the virus, understanding the host immune response in particular can help prepare us against the continuous evolution of the virus, where new variants of concern or VOC are being reported from time to time. Herein, we identified highly conserved HLA supertype restricted T-cell epitopes that capture the diversity of the viral variants and are applicable to the human population at large. All reported sequences of the structural proteins, which are spike, envelope, membrane, and nucleocapsid, were downloaded from the GISAID EPICode database as of January 2021. The sequences were processed for each protein and duplicated using CD-HIT and aligned with muscle. Table 1 shows the number of redundant and non-redundant sequences of the 4 proteins. These are the proteins that we studied. And in this second column, we have the number of redundant sequences. The total numbers were more or less the same across the protein, about 400 key, resulting in a total of slightly more than 1.6 million. In this third column, provides the number of non-redundant sequences after removal of exact duplicates. The number reduced drastically, ranging from 85% to close to 100%. The non-redundant numbers for each protein were not similar. For example, 60 key for spike to 1 key for envelope, giving us a total of 87 key. Our analysis focused on the study of only full-length sequences, which totaled close to the 80 key. Given the large number of sequences for some of the proteins and for consistency, the partial sequences were filtered out to avoid spurious alignments. Shannon entropy was used to quantify the protein sequence diversity by use of ABK-ABANA. An overlapping chimer window of 9 was used for immunological applications. The entropy plot is shown in here, figure 1. The entropy pattern between the protein is different. The higher the entropy value, the more diverse the monomer position with entropy of 0 indicating complete conservation. In general, the envelope protein had a higher entropy. However, the peak was observed in nucleocapsid protein, which was close to a value of 1.8. The spike protein, which has the center of attention, was generally of low entropy, with few positions of high diversity. Overall, the structural proteins were generally conserved, with a low mean monomer entropy of approximately 0.13. Highly conserved monomers, defined as position of entropy lower than 0.4, were determined and concatenated if they overlapped, as shown here in Table 2. There were more than 1.7 key highly conserved monomers, with the most from spike. And they concatenated to 14 sequences. These 14 concatenated sequences were selected for the prediction of HLA-A2, A3, and B7 supertype restricted epitopes by use of net-MHC cons. Since the tool predicts for the individual alleles, a criteria was set to identify supertype restricted putative epitopes. The 3 HLA class 1 supertypes studied alone provide for a large population coverage, approximately 86%. The prediction results are shown in here, Table 3. 33 putative epitopes were predicted within 7 of the concatenated sequences. As we can see here, only these 7 had a positive prediction result, Such putative epitopes were validated by comparison with experimentally confirmed epitopes reported in the immune epitope database. In conclusion, these highly conserved sequences, specifically these 7 shown in Table 3, are potentially at the interface of interaction between the human immune system and the virus, and they merit further investigation as epitope-based vaccine candidates that capture the diversity of the virus and are applicable to the human population at large. This is a preliminary study to something larger where we will be expanding the work to all other proteins of SARS-CoV-2. Thank you.